there will be additionally CD8. These are the marker of the T lymphocyte, and the major important marker of B lymphocyte is CD19, CD20 and CD21. So this is very important, you have to remember, because you will have this question related to this. Okay, so B lymphocyte, a cell with a big nucleus, very small cytoplasmic membrane, and they have the surface marker, no function until activated in the secondary lymphoid tissue. Once activated, they will convert into the plasma cell. B cell will convert into the plasma cell and form antibody. T cell is the organizer of all this immune system. They will activate, and then they will activate the B cell and form the this antibody. So these are the organizer. They reorganize everything, they manipulate everything, but they manipulate the B cell for formation of the antibodies. So they are actually manager, you can say. Then coming to the plasma cell, this is the activated form of the B cell, the blood stream, secondary lymphoid tissue and bone marrow, small eccentric nucleus with intensely staining Golgi apparatus. So this is, you can see the same cell, this cell has converted into this. After activation, the nucleus has shrink down into the periphery, and there is the Golgi apparatus over there. So this is your plasma cell, the intensely staining Golgi apparatus, terminally differentiated of the B lymphocyte that secretes the antibody. So you can identify the plasma cell as well. Now come to the natural killer cell, and natural killer cell is present in the blood stream, the lymphocyte with the large cytoplasmic granule. There will be the granule will be more prominent, and their surface marker is important. I have told you all lymphocyte surface marker is important. This is CD16 and CD56. So CD16 and CD56 is the surface marker, is the identifying marker of your natural killer cell, which is kills the virus and tumor infected cell. Okay, they kills virus infected cell and tumor cells. So this is the function of the NK cell. They came from the lymphoid progenitor cell, but they are not part of the adaptive immunity. They are part of the innate immunity. So this is also very much important. Now coming to the white blood cell, coming about the monocyte. The monocyte is found in the blood. The same monocyte, if comes in the periphery, it becomes the macrophage. The monocyte is circulating in the blood. They are a granulocyte with bean shape or kidney shape nucleus. That is important. They have this nucleus which is bean shape or kidney shape nucleus. So this is the characteristic marker of your monocyte, which is another name of macrophage. If it is present in the blood, it is monocyte. If it come outside, it's become the macrophage. These are the precursor of tissue macrophage. So these are the precursor actually. Okay. Now talking about the macrophages. Macrophages actually are resident in all tissue. This is, you can see, this is the macrophage, a granulocyte with ruffled cytoplasm. So cytoplasm, rough cytoplasmic membrane. So this, you can see this membrane. This membrane is actually irregular. That is not uniform like monocyte. This is or you can say circular monocyte, but here the macrophage is zigzag. That is important. Resident in all tissue, ruffled cytoplasmic membrane and cytoplasmic vacuole and vesicle. So they have cytoplasmic this vacuole and vesicle, and this has the ruffled cytoplasmic membrane. Okay, and these have the function like phagocyte, prophylogenic antigen presenting cell and T cell activator. So they have other function as well, like they have macrophage is mainly responsible for phagocytic cell. They can also act as your antigen presenting cell, and also they are T cell activator. So you need to now understand, you have actually understood that monocyte in the blood is, when it comes into the periphery, it sends the cytoplasmic membrane becomes ruffled. They contains this nucleus, but nucleus is shifted towards one end, and there are the vacuoles and vesicles inside the that macrophage. So this is you can understand. This is the phagocytic cell. They can present as antigen presenting cell. They can also act as a T cell activator. You should understand, to understand the immunology, macrophage is one of the major role player. So macrophage is actually antigen presenting cell that present all your antigen, and this that protein in nature component is processed by it and then goes to the T cell, that activate the T cell, then activate that T cell will activate the B cell and form the antibody. Okay, now coming to the dendritic cell. Dendritic cell is resident in the epithelial and lymphoid tissue. They are not present everywhere. They are present in the epithelial lymphoid tissue. They are a granulocyte within thin satellite cytoplasmic projection. So you can see the projection is like zigzag. It has not a uniform projection. This is called satellite cytoplasmic position. It is also phagocytic cell. It is also antigen presenting cell, and it is also T cell activator, same as the macrophage. So it is dendritic cell that is same as a macrophage's function, and it is also a antigen presenting cell. So the antigen presenting cell in the body is macrophage, dendritic cell and B cell. So these all have this MSC class 2 protein, and all the cells have MSC class 1. including this antigen present in macrophage and dendritic cell as well. So now coming to the eosinophil, you can see this is the bilobe nucleus with the large granules. This is circulating blood cell recruiting into the loose connective tissue of the respiratory and GI tracts. They are granule site with the bilobe nucleus and large pink cytoplasmic granule. So they are the bilobe nucleus with the large cytoplasmic granules. Elimination of large extracellular parasite and type 1 hypersensitivity. So their function is elimination of the large extracellular parasite and as well as type 1 hypersensitivity. And coming to the mast cell reside in the most of the adjacent tissue to the blood vessel. Granulocyte with a small nucleus and large blue cytoplasmic granules. Elimination of large extracellular parasite, type 1 hypersensitivity. So this eosinophil and basophil actually, they both are looks like the similar. They also are the bilobe nucleus. This is also the bilobe nucleus. Granules with the bilobe nucleus, but they are large blue cytoplasmic granule. So they have the blue cytoplasmic granule and these have the pink cytoplasmic granule. Okay, so basophil, eosinophil and basophil both are bilobe nucleus, both are similar in size. They have cytoplasmic granules that is pink in color. They have cytoplasmic granules that is blue in color. They are all responsible for the elimination of extracellular parasite and type 1 hypersensitivity. This is also responsible for extracellular parasite, elimination of extracellular parasite and type 1 hypersensitivity. The mast cell has this structure. This is similar to the basophil. They are reside in the most adjacent tissue and near the blood vessel. So when mast cell degranulates, the histamine is released that goes to your blood vessel, cause dilation, and you patient goes into the shock or say BP falls down. So this is of your mast cell. Laboratory evaluation of the patient commonly involves assessment of the white blood cell morphology, cell count by examination of blood sample. Changes in the morphology and portion of the white blood cell indicates the presence of some pathological state. So these are the normal structure, and if there is any changes in that, then we can state there is some pathological state. A white blood cell, this is also important. A white blood cell standard blood count includes neutrophil, band cell, lymphocyte, that is T lymphocyte, B lymphocyte, NK cell, monocyte, eosinophil and basophils. So you can see in the subsequent lecture over here. So here in the subsequent slide you can see this is the neutrophil is 70 to 80%. So you can see 50 to 70% there is a neutrophil. Then band cell is 0 to 5%. Then there is the lymphocyte that is 20 to 40%. There is the monocyte that is 5 to 10%. There is the eosinophil that is 0 to 5%, and there is basophil that is 1%. This is your differential count of your peripheral blood smear when you do WBC differential count. So you have to do neutrophil, you have seen the structure, you have to see band cell as well, lymphocyte also, monocyte, eosinophil and basophil. So this is all about actually our chapter 2nd. We will discuss our chapter 3rd in subsequent lecture. Thank you.